Ladies and gentlemen, welcome to our biology class, and in this class we're just going to look at MCQ biology revision, of which we look at 2022/2023 biology 1400 exam solution. So in the previous class we discussed question 1 to 25, and this time around we are going to look at question 26 to 50. So if you have any question, feel free to ask. For those who might have questions after the class, you're also free to inbox me in my inbox. Okay, so with that being said, we can quickly start our class by looking at question 26. The molecule below is dash. So this diagram is in your notes, and this diagram is also in our video where I was explaining about carbohydrates. Specifically, this has to come from polysaccharides. So go and check out that video on carbohydrate, which is lecture part 3. You will find the exact diagram. So if you look at the nature of this guy, one thing you need to understand is what are these subunits. So if you look at these subunits are just glucose. This is glucose. And then if you look at the naming, you can find that this has to be on 1, then there are 2, then 3, then 4. Yeah. And then if you look at this one, it's having 1 there, 2, 3, 4. So mostly this is a 1,4 glycosidic bond. So this is a 1,4 glycosidic bond. That's the first thing you need to identify. So after that, you have to check up the molecules, which is here. Automatically it can't be fructose, it can be fructose in this case, but one compound which can be this one, which is a polysaccharide, it is just amylopectin. Yeah, amylopectin. So the answer here is amylopectin with 1 alpha 1,4 linkage of glucose. So make sure that you do your research on that, and you can also check the lecture notes and also the lecture video which I posted on our YouTube channel. We can move on and look at number 25. Identify the correct statement about fats. So this question is coming from lipids. This question is just coming from lipids. So the first option is saying saturated fats have straight chains and are solid at room temperature. So which statement is correct? I can't even go down there because I already know that the saturated fats are the ones which have no double bond. The saturated fats are the one which have no double bond, and them not having a double bond, they don't have a kink structure. So they don't have a kink structure, no kink. Okay, they don't have a kink structure. This kink structure is just a bend which you have to see in the saturated fat, in the saturated lipids. And them not having a kink structure and them appearing in a straight chain, they normally tend to be so much compacted together. So if the molecules are closely packed, then that simply means that these guys have to be solids at room temperature. Unlike the unsaturated fat acid, these ones, they normally have a bend and they are liquid at room temperature because these molecules are not closely packed. The molecules are not closely packed because they have a bend. As a result, these guys, they tend to be liquids at room temperature. Okay, let me quickly go to number 26. Number 26 is saying, which of the following statement is false about hydrogen bonds? So this question is coming from water, and you can also talk about this same question on type of bonds. So we are looking at the statement which is false about the hydrogen bonds, and if you look at the option, they are responsible for the high boiling point of water. This is true, guys. This is true because of the fact that these hydrogen bonds need to be broken down for water to evaporate. Then they are a weaker than van der Waals forces. If you compare very well, and if you remember from the lecture class, actually I explained the difference between the hydrogen bonds and the van der Waals forces, and I said that the hydrogen bonds are stronger than the van der Waals forces. So the answer here, it is this one, which is false, because of the fact that the van der Waals forces do not have a strong bond as compared to the hydrogen bond. Then they are weaker than covalent bond. It is true. And then they are weaker than ionic, which is true. They are responsible for holding the two DNA strand, which is also true because of these guys, the helix structure. So that helix structure in DNA is due to the presence of the hydrogen bond. So this is it for this question. The most common lipids in cells are, okay, so some of you might have not yet done the lipids, but I'll try hard to explain the lipids. So the lipids, these are also biomolecule. These are also biomolecules, and they have a lot of functions in the body. One of them, they act as enzymes. They act as hormones, not enzymes. And then they play a role in structural support. And they can also play a role in the provision of energy. For example, if you look at a triglyceride, which is the most common guy in most of the cells. But yeah, so in this case, the most common lipid in cells are just the phospholipids. The most common lipid in cell is just the phospholipid. You need to understand from our part 1 of this revision, which we did from question 1 to 25. If you remember very well, we looked at the cell membrane having the phospholipid bilayer. I remember explaining about that, and also this cell membrane also having the lipids called the cholesterol. Yeah, the steroids, these are hormones. These are hormones. Okay, you know much in detail about them when you study biochemistry. Cholesterol is found in the cell membrane, this guy here, and this guy is formed by the what? Fat acid and an alcohol called glyceride. And then glycolipids, it's just a combination of carbohydrates and the lipids. And this one, it is found in the cell, or sometimes it is there. But the answer for this guy is just the phospholipid, which is found in the cell membrane. So if you look at the structure of the cell membrane, it has a lipid bilayer, and this lipid bilayer, it is made up of phospholipid. With which of the following substance can lipid form a micelle? Okay, I think on the topic of water from our tutorial video, from our tutorial lecture, if you watch it very well, I talked about certain substances such as lipids are immiscible in water, and as a result they form what we are calling a micelle, which is found just there around. So the answer here is just water. So in water, mostly the lipids, they are able to form a micelle substance. And the reason why they are able to form the micelle substance is because they are insoluble in water. They are insoluble in water, but they are able to interact with water. They are able to interact with water. So they cannot dissolve in water, but they are able to interact in water, such that they have this as the head and this as the tail. Okay, I'm just indicating that as the tail. Okay, so this is the head. Okay, this is the head and this is the tail. Sorry, I've put the head on the tail. Okay, so they have the head and the tail. They have the head and the tail. So this is the head, this is the head, and then this head, we also have the tail. Okay, so when you talk of the tail, the tail is hydrophobic. The tail is hydrophobic. For the explanation of the word hydrophobic, you can watch the tutorial lecture class on water. I explained what hydrophobic mean, which is being insoluble in water. Then the head is hydrophilic, which is water loving. Hydrophilic, which is water loving, and these guys, they are able to interact with water, whereas this guy is not able to interact with water. Then overall, this guy is said to be amphipathic. Amphipathic. I hope my spelling is correct, because it's not able to dissolve in water and it's also not able to be insoluble, but it's in between the two guys. Okay, with that explanation, I can move on and look at the next one. Which of the following fat acids is monounsaturated? Which of the following fat acid is monounsaturated? So the word monounsaturated, it simply means that it has one double bond. Okay, it has one double bond. And when you talk of unsaturated fat acid, unsaturated fat acid, you need to understand that this unsaturated fat acid one, they have at least a single bond, a single double bond, at least one single double bond, double bond. And then another thing you need to understand about these guys is that they are bent. Okay, they are bent, and this means that they form a kink structure. They form a kink structure. So them forming a kink structure, they are spacely packed. that simply means that they are spacely packed, okay, because of that king structure. So they are spacely packed. So when they are spacely packed, this also simply means that they are liquid at room temperature. They are liquid at room temperature. I think I'll explain more about these lipids. I'm going to arrange a class for you guys specifically about lipids and go in detail about them so that you guys can be helped and understood. So for this question, the answer is simply for this question, the answer is simply let me go through. We have C7H32O2. So the answer is just 5. Yeah. But I'll explain more about this. I'll explain more about this when we do lipids. I'm going to teach this topic within this week. Let me move on and look at the next question. Some insects can walk on water. Okay, for this one, the lecture class is already there. Go and watch bio lecture class, and specifically on water. And of course, this will deal with the hydrogen bond, and this will also deal with the high surface tension. So like I do in mathematics, before I attempt the answers, I first try to look at the topic and what the question is trying to focus on. So for this one, it's coming from water, and then they'll talk about hydrogen bond and the high surface tension. So in that case, the solution for this particular question is simply let's say some insects can walk on water because one of the hydrophobic forces. No, this cannot be correct because of the word hydrophobic. Of adhesive forces between water, this is also wrong because of the word adhesive. Of cohesive forces between water, okay, this one I'll put it on a pause. And try to compare it with other options. I won't really go fast and pick it up, but I'll put it on a pause. Water is electrically neutral. No. Of cohesive forces between water molecule and the insect. No. This has to be adhesive. Forces. So the information in each and every student, the information which you guys would want to hear from this question, it is the issue of what I just stated, which is the hydrogen bond and the surface tension. But yes, these things have been mentioned in this same solution, which is 3 as our answer, by making them enter the term cohesive force. Because if you have a water molecule, this is oxygen, that is hydrogen, okay, and another hydrogen there, then you have let's say oxygen there, hydrogen there, and then you have hydrogen there. Remember that this guy actually, this is not supposed to be close because the forces need to repel the guys there. The forces need to repel. So this one, hydrogen there, okay, that one also hydrogen there. Okay, because of the difference in electronegativity, ladies and gentlemen, we are revising, so expect me to be summarizing everything. So because of the difference in electronegativity, so electronegativity is just the pull of electron towards an element, or the ability of an element to pull electron toward itself. Or electronegativity can be said to be the ability of an element to attain the negative charge, So because these guys, they do normally share the electron, so in such a case, you need to expect this one to be partially positive, that one to be partially positive, that one to be partially positive, that one to be partially positive. Then this other guy will be partially negative, that also will be partially negative. Now, what will happen is that there is a bond which exists between these two molecules, like that. Okay, again, this oxygen is capable of making another bond. Okay, then automatically this guy will also be linked to another water molecule. It will also be linked to another water molecule. There is a reason why I want to write this. Okay, so this is partially positive, and that one partially negative. And then the other thing that you need to check out is these are the HB bond, and these bonds are making a cohesive force. So the force which is between the two water molecule is a cohesive force which is existing between like molecules, Yeah, so for more information, check out my tutorial video, my lecture class on water. Identify the correct chemical formula for glycerol. So glycerol is an alcohol. And this alcohol takes part in the formation of triglyceride. So I don't really need to go much in detail because this formula is in your books, so you just have to go and search and look for the formula. So this is how it is, like that. So that's the final answer for glycerol. Okay, with that being discussed, we can move on and look at the next guy. Identify the incorrect statement about amino acid. So amino acids are just monomers of proteins. They are just monomers of proteins. Okay, they are just monomers of proteins, and these guys, they do connect to each other to form peptides. Which are just proteins. So they have the same basic structure. That's the first option. And then the second option is saying each amino acid has a unique aryl group. So the basic structure is correct. If you check in your notes, this is true. Each amino acid has a unique aryl group, which is also true. They are amphoteric molecule, which is true. So them being amphoteric molecules, that simply means that they are able to control the pH of the what? The pH of the environment. Let's say the pH of a cell, it's being controlled by these amino acids or proteins. So if you look at that, because of them, if the pH is high, let's say it is alkaline, these proteins, they will act as these amino acids, they will act as what? They will act as acid, so that it can be neutralized. And then in a case where the pH is low, they'll act as alkalines. Then for the structure here, I think I have to explain the structure there. For protein is such that you have a C there and then H there, sorry, for amino acids, then you have the R there, then you have the NH3 positive, and then you have a COO negative. So that's the basic structure of the amino acid. All the amino acids have this structure, okay? All the amino acids have this structure. And then for this structure, what you need to understand is this part here is known as the carboxyl group. This is known as the carboxyl group. Take note of that. And then this one is known as the amino group. This is known as the amino group. You should be able to know the label of these guys here. Then this one is known as the R group. And this R group is where the difference is. This R group is the one which cause the distinction, So what I mean is it's the one which makes one amino acid to be different from another. So that makes 1 and 2 to be correct statement. This one is also correct. Then the R group are involved in peptide bond formation. This is the wrong question, and this is the one which is correct. So since we are looking for the incorrect, so this is the answer. So the guys which take part in the peptide bond, which is formed by a condensation reaction, since water is produced, the guys that are involved are simply the carboxyl group and the amino group. These are the two which are involved. Which of the following options represents the ground state electronic configuration? So one thing that you have to understand is when you talk about the ground state electronic configuration, is just the arrangement of electron around the nucleus of an atom with lower energy level. So the one with lower energy level. So here it is. So if you look at everything here, though I can't see, this a 3 and that's also a 3. So let me analyze everything. So this is 2, 4, 7 plus 3. Okay. So for this question, what they are just trying to demonstrate here is to help us to know if at all we know how to do the electron configuration. If at all you know how to do the electron configuration. So I'll start with the first one. The first one is wrong because there is a 7 there. This 7 is not supposed to be there. Instead, you need to have a 6. Okay, so this one is wrong. Then I'll go for this one. 2, 2 is okay. 5, because of a 5 there, it is wrong. This is supposed to be 6. Okay, so that 6 plus 2, it will be 8. The rule is that do not go to the next level without filling up the other level. Then 2, 2 is okay. 6, 2. This one, I'll pause a bit. It is correct, but I don't just have to rush and pick it as the answer. It is correct, but I don't have to pick it as the answer. Instead, I have to chill a bit. This one is wrong because there is supposed to be 6. This one is wrong because there is supposed to be a 2. There is supposed to be a 2. So in this question, they were just trying to see if you know the number of electrons that are being occupied in the first shell, second shell, and also the third shell, and also helping to analyze if you're able to know if how many electrons are supposed to be in one single shell. So there, the answer is simply this one. Okay, so for this and that, together with that, these three, they were trying to ask you how many electrons are in the p orbital, because there are normally 3 p orbitals. Okay, you have 3 p orbitals. So since we have 3 p orbitals and each has a maximum of 2, so if you say 2, 1, 2, 3, and then this is px, this is py, and this is pz, then in that case I'll pick this one alone. Then you have 1s there, then you have 3 which has s alone. So for that, you have to understand that there it will be 2, a maximum of 2. Remember here there has to be a 2s. That one also 2. Okay, they have to be in opposite direction. Those 2, those 2, those 2, and then next it will be what? It will be 2. Yeah, so here it has to be 6, maximum of 6. There it has to be maximum of 2, there maximum of 2, and there maximum of 2. So that's what this question was just trying to be analyzed. Which part determine the basic characteristics of an amino acid? Which part determines the basic characteristics of amino acid? So guys, these things is all about understanding. Okay, it's all about understanding and making sure that you go through your notes. So if you look at this question, one term which is important is 1, the basic there, 2, amino acids. Okay, So when you understand what you are trying to talk about, what the question is trying to talk about, then you'll be able to understand the solution. So the basic character of an amino acid, basic character, it's not talking about the overall character of an amino acid, okay? It's talking about the alkaline side of an amino acid, the alkaline, okay? So we said this is the central, this is the R group, this is the H group, this is COOH, yeah, COOH, and then there I'll write this, the carboxyl group. You have the carboxyl group, and then you have the amino group. So this one, it's determining the basic nature, the basic character, okay, of an amino acid. Sometimes they'll ask you which one makes the amino acid to be basic. And then this one, it's the acidic nature of an amino acid, the acidic nature of an amino acid. So here the answer was simply 4. Okay, so before I proceed, do we have people with questions starting from the first parts up to the last parts? Do we have questions, ladies and gentlemen? If you have a question, you can raise your hand. Okay, I have not given much chance of you guys to ask questions because I am a little bit going a bit in detail so that I can highlight a lot of points, but most of these things are in the lecture notes, and most of them are also in the classes which I have already posted on our YouTube channel. Okay, no question? We are good? If we are good, what is the name of this bond here? What is the name? If we are good, what is the name of the bond? What is the name of that bond which I have pointed there? Anyone to try? Frederick, what's the name of that bond? Yes, Nelson.
Is it the hydrogen bond?
Not really. The hydrogen bond is the one which is dashed. Yes, Nelson.
Covalent bond.
Okay, covalent bond, correct. But it's not covalent bond. It is a polar covalent bond. Polar covalent bond. We have two types.
That's what I said. I said the polar covalent bond.
Okay, congratulations for the first time. So polar covalent bond.
That's what I said.
Polar covalent bond. That's the type of bond which is there. Okay, good that you said that. Okay, let me move on and look at the next part. Which of the following bonds are not present in the tertiary structure of proteins? It's a very nice and interesting question. I think this question has been repeated like 3 times. I've seen it in some other papers. So which bond is not in the structure of a tertiary protein? Okay, the first one, the first thing you need to tick is the hydrogen bond. It's there. Disulfide, it is there. Ionic is there. Peptide bond is there. The one which is not there, it is the phosphodiester bond. The phosphodiester bond, you find them in the nucleic acids. You find them in the nucleic acids. So these bonds are not found in the tertiary structure of a protein. This tertiary structure of a protein is also said to be the functional level, or the functional structure of proteins. Okay, and this is where now a protein has already achieved a 3D view or structure, a 3D structure. Yeah, at this particular point, a protein has attained its 3D structure. As a result, it becomes functional and it's able to do its functions. It's able to do its functions at this particular point. Any change in the 3D structure of a protein, it makes its physical and chemical characteristics to be changed. For example, if you get, let's say, which one can I get? I think beans. Yeah, let's say beans. Okay, so if you eat the uncooked beans, and you eat a cooked beans, you find that there is a difference in terms of taste. There is a big difference in terms of taste when you eat the uncooked beans and the cooked beans. Why? The uncooked beans, the structure of the uncooked beans is different from the structure of the cooked beans. So why the structures been denatured and deformed? A protein with 4 peptide bond is called, wow, this is a very easy question that anyone can get right. So it's just the penta, peptide. Yeah, that's the answer. Okay, we move. Substances that are inactive in the action of enzymes by binding to enzyme allosteric sites are referred to. So that topic is enzyme, and this is just asking for the definition, and the answer here is just 3, which is non-competitive inhibitors. Which one of the following statement is correct? Which one of the following statement is correct? So this one is talking about the atomic structure. So this question is from atomic structure. So if you view electron close to the nucleus have no energy, this is wrong. Electron closer to the nucleus have higher energy, which is wrong. Electron further away from the nucleus have lower energy, which is wrong. Guys, even if you don't understand, you can see higher, and then this one is closer, away, and then this is lower. Automatically you can see if you pick this one as your answer, then even that one is your answer, because these are opposites of each other, right? Because you have the nucleus there, you have the electrons in this first shell, okay, you have electron in the first shell, you also have the electrons in the last shell, okay. And one thing you need to understand is these electrons have different energy. Like this one, these are what? Valence electron. These are valence electrons. Like in this case, these are valence electrons. And then these ones are called the core electrons. The core electrons, okay. So if you analyze closely, these guys have lower energy, okay. These guys have lower energy. Yeah, these guys have lower energy. That's why they are close to the nucleus. During a chemical reaction, they won't take part in that chemical reaction because they are very close to the nucleus. These guys have higher energy. They are a bit excited, higher energy. They are a bit excited and they are ready to do any chemical bonding. So electrons further away from the nucleus have higher energy. So this question was very simple. So the answer here is just 4, which is what I've shaded. We move the folding of polypeptide chain into beta plated sheets results in a structure dash. Okay, this one again, it is coming from protein, so this part can also be given alpha plated sheet. So secondary proteins, when you talk of secondary proteins, at this particular stage, the proteins tends to fold. The protein tends to fold. Actually, it's not really the proteins, but we can use the word the primary proteins. So the primary proteins tends to fold, and the bond which is existing, it is the hydrogen bond. It is the hydrogen bond. So the secondary proteins, which is the answer in this case, they are the ones that have a hydrogen bond in them, and these guys, they produce a beta plated sheet and the alpha plated sheet. An example of a secondary protein can be keratin. Yeah, you can pick up keratin, which is found in hair. So this one is an example of a secondary proteins. Okay, which of the following statement about enzyme is false? Which of the following statement about enzyme is false? So you have enzyme are protein in nature. Yes, this is true. Enzymes are protein in nature. It is very true. Then each enzyme catalyzes one specific substrate. Yes, it is true. Amylase can only catalyze amylose, and then cellulase can only catalyze cellulose. So there is no other way around. Then a temperature above that can denature human enzymes. This is correct. Denatured enzyme can catalyze a chemical reaction at a lower rate. This is wrong. Once an enzyme is denatured, it loses its function. Which of the following pyrimidine is found in both RNA and DNA? which of the following pyrimidine is found in both RNA and DNA? So here the answer is just cytosine. So cytosine is the one which is a pyrimidine. Which of the following biological molecule is represented by the general formula that? Okay, so basically guys, if you go and check the lecture class on carbohydrates, specifically lecture number 1, lecture part 1, you are going to understand that this is the basic formula of monosaccharide. So the answer here is simply glucose, because this one will lose some H2O, there is loss of H2O, this one there is loss of H2O, this one there is loss of H2O, okay, H2O lost, H2O lost, H2O lost, and this one no oxygen. Okay, is it no oxygen? Ribose, deoxyribose. No, this one is a what? Is a pentose sugar. It's a pentose sugar, but basically the answer has to be this one, which is correct. Genes which are present in the homologous region of X and Y chromosome are. I think I'm not going to do genetics. I'm not going to do genetics, but I'll send you a class which has genetics in it. I'll send you a class which has genetics in it. So I'll explain this one later on, because this is now going to genetic side. Which of the following structure is not found in plant cells? Okay, this one is molecular. So which one is not found in plant cells? So the answer here is 4, which is the centriole. Okay, the centriole are only found in animal cells. Out of the 20 amino acids, only dash is optically inactive because it contains dash. So the answer to this one is 2, which is glycine is because of the hydrogen. So where there is the ara group in glycine, in glycine, you have what? You have hydrogen. This means that it is optically inactive, because for an object to be optically inactive, it means that no chiral carbon. So for the concepts about this, you can check out my notes on carbohydrates, where I talked about stereoisomers. Yeah, so that's the part. Choose the first statement about the structure of RNA. So here thymine is part of RNA structure. This is wrong. Thymine is found in DNA. So in the meantime, for those with questions, I'll give you time to ask, and for those who might want to ask questions later on, you can inbox me on my WhatsApp line. So for now, I'll allow you guys to ask questions.